As my research program evolved in the context of a very rapidly growing field of tissue engineering, I realized that regenerative medicine, or replacing organs and tissues in the body, was not necessarily the best application of tissue engineering. One huge gap in our ability to develop drugs and therapies and even to understand disease are better complex models of how human tissues and organs behave that we can manipulate in culture. So mouse models and other rodent and animal models only take us so far. Right now, it is very challenging to get a drug from preclinical studies all the way through clinical trials, because very often drugs are toxic or have off-target side effects that can't be predicted in the kinds of studies we now do in animals and other preclinical predictive studies. So about 10 years ago, we began to focus tremendous efforts on building 3D models that capture in vitro capture complex human physiology in vitro, particularly 3D tissues, and in particular in liver, for example, recognizing that these tissues are perfused at a micro scale with fluids that provide nutrients but also provide mechanical stresses. So we've led development of a very simple to use 3D microplate version of a liver, now in development with several pharmaceutical companies, that allows you to test the effects of drugs and biologics on liver tissue. And there are the challenges for the future that we're facing are, it's one thing to build these kind of tissues, it's quite another to get useful predictive information for drug development out of these. So a huge focus right now is not just the kinds of making the tissues that we do in our lab and others at MIT and elsewhere do, but really how do you interface with the systems biology community to get useful information out? How can you do the fewest possible experiments to get the most information by driving these kinds of systems into the kinds of pathologic situations you might encounter in vivo, for example. So we've teamed up in a number of different ways with people in the cancer biology community and other systems biologies communities at here and in other institutions to try to link tissue engineering to systems biology in order to provide a more comprehensive way to think about these very huge challenges in understanding diseases and in developing drugs. One example of how we're doing this for challenging diseases is working together with collaborators at the University of Pittsburgh and here in MIT's Koch Institute. We are trying to understand how a single cell metastasis in liver can go from being dormant to being activated to grow into a micrometastasis and ultimately into a observable liver tumor metastasis. As we know, it is very often the metastases that kill patients and not the primary tumors. So we're trying to understand the process of escape from dormancy by using complex 3D models in ways that we might ameliorate or prevent that by giving patients, for example, drugs that may not have tremendous side effects but may hold in check the kind of events that would allow escape from dormancy. That's in very early stages, but it's an area, again, where all of these systems biology methods can yield tremendous insights when coupled with facile, easy-to-use tissue engineering approaches. A second area of emphasis right now is and developed over the past 2 or 3 years, is a focus on women's reproductive health. In particular, things that involve disorders of the uterus and other things that are gynecology related. In fact, we founded a center here at MIT, the Center for Gynepathology Research, which now includes about 10 investigators and has about $2 to $3 million a year of funding in foundation and government support to focus on bringing new kinds of approaches to studying endometriosis, adenomyosis, and other very common, highly morbid disorders that women face, particularly young women, things that derail their professional careers. So we work closely with clinicians at Harvard Medical School who operate on these patients and treat these patients to study patient samples and develop platforms for drug discovery and also to understand the etiology of these very perplexing kinds of diseases that are poorly understood. in these women's health projects, again, a focus is combining systems biology with tissue engineering. So on the one hand, for example, we're doing a comprehensive analysis of what's in the peritoneal fluid of women who have endometriosis and women who do not have endometriosis and present for other symptoms. That includes analysis of cytokines, cells, and other small molecules in the peritoneal fluid during various stages of the menstrual cycle. At the same time, we're building tissue engineering models of the endometrium and other aspects of the uterine lining in order to understand how the eutopic tissue contributes to formation of these ectopic lesions and whether it contributes. We also are exploring whether stem cells are contributing to this disease by linking some of the studies that we've had for many years in bone marrow stem cell biology with some biology that may be occurring in the peritoneal cavity in women with endometriosis.